story short,
So he can progress it. If he controls it. Maybe you can use Spotlight for all of us. Spotlight his camera. I think his video's on, no? There we go. Yeah. You can hear us? Hi, Saurav.
Yeah, I can hear you. I am sharing my screen at the moment. I'm not sure if you guys can see it.
You can see it. It says loading, though. It's a black screen saying loading.
Oh, okay. Let me.
Maybe you can refresh.
Again. Is it coming up now?
I see a black screen. I don't know if that's your home screen, but
let me try this again.
yeah, we see that there.
Yeah, so it's
Something happens when he presses.
Yeah, I want to talk about is
Okay, so maybe if I
Can control on our end if you want, but I need to.
okay. Let me just try to.
Okay, that looks good.
All right.
Yep, you're all set.
All right. So I can start now.
Yeah, go ahead.
All right. Good morning, everyone, and thank you for joining me here today. I'm really excited to be part of the Knowledge Graph conference, although I didn't get to attend it in person this year. My name is Saurav, and I lead the engineering team at Datum. To introduce Datum briefly, it is a startup which is building a connected health platform that brings together biomedical knowledge and healthcare data to enable applications in precision medicine and decision support. So let me get started with the presentation. So here's the basic outline of the topics I'm going to cover today. I'm going to start by talking about why it is still so hard to build applications in the healthcare domain. And to make the context more concrete, I'll be introducing a use case for pharmacogenomics, which is an important component in precision medicine. I'll then go through the challenges developers and researchers face day to day when developing application for precision medicine. And in the process, introduce a solution that we've been building at Datum for the past few months to work on these challenges. So let's first talk about the sources of complexity in developing healthcare application today. It's no secret that healthcare is a data-rich domain. And if we can fully expect that the volume and the variety of healthcare data is only going to grow exponentially in the future. But despite this, healthcare data is largely untapped resource and has not been harnessed adequately. There are many reasons for this, but the fact that the information in these sources are very specific to healthcare domain is a big factor. And because of that, building platforms to integrate this domain-specific knowledge into these applications is still a complex undertaking that most engineering teams are not able to uh, develop easily. This also makes generating insights for this data out of reach for most teams, as it requires building bespoke data pipelines and data models. So extracting value from biomedical data generally requires a multidisciplinary team of bioinformaticians, data scientists, and data engineers that collaborate closely with the research scientists who are the domain experts. But despite these efforts, most researchers and practitioners are only able to extract and analyze a small fraction of this data. Healthcare is also a highly regulated domain, which means it is governed by strict data privacy, fraud prevention, and security requirements that most of other domains don't necessarily have. So, for any healthcare application involving patient data to be deployed in a real-world setting, it needs to conform to an ever-increasing list of regulations that govern this industry. So, developing regulatory compliance expertise is a must-have for successful healthcare deployments. However, setting up these interdisciplinary teams is quite time and resource intensive process, and only a handful of large institutions are really capable of doing this at scale. And a lot of the same challenges are also present on the developer side of things. The patient data is generally spread across a number of different systems, and each of these systems can have their own data formats and follow similar but slightly different schemas, ontologies, and vocabularies. This means a lot of effort is required to reconcile data from these systems into a single coherent data model. Let's talk about a more specific use case in healthcare that our team has been working on, which is pharmacogenomics. I'm sure like a lot of us have not heard about this particular term before, but in very simple terms, pharmacogenomics or PGx is the study of how an individual's genetic makeup influences their response to a particular drug and or combination of drug. I've already mentioned that it is an important field of precision medicine. And precision medicine itself is a more general approach to disease treatment and prevention. It takes into account other factors like a patient diet, environment, lifestyle, demographics, in addition to the genetic profile that we are interested in pharmacogenomics. So to give some intuition on what pharmacogenomics is all about, let us take an example of a patient who is being prescribed a drug by a clinician. From a pharmacogenomics point of view, when a patient takes a drug, we normally expect that over time, the patient drug concentration in their body stays within what is called a therapeutic window. So each time they take a certain dose of the drug, the concentration will go up, and over time they'll metabolize some of it away until the next dose when the concentration increases again. Um, uh, But for certain other patients with a different genetic profile, like the one indicated by the red line here, even if they are given the same dosage of the same drug, they might not metabolize it quite as well. So over time, beer levels might build up and hence they could experience adverse effects from taking the drugs. On the other side of things, some patients, like shown in the purple line here, metabolize the drug a lot faster than the average patient. And in this patient, their drug concentration might be too low. And as a result, the drug will stop having any effect in the patient over time. And these differences in metabolic reactions of the body to drugs are said to have different drug phenotypes in general genomic parlance. And to ensure normal metabolization, the patient should be prescribed different dosages of the same drugs or even alternate drugs. An example of this can be seen in this table here, which is taken from a pharmacogenomics guideline publication for a particular drug and a gene pair. If you look at it closely, for a poor metabolizer of the drug, the recommendation is not to prescribe that particular drug in question due to the increased risk of certain diseases in the patient. While in the opposite case, for the rapid metabolizer, since the drug will not have any effect on the patient long term, there are no recommendation for that drug. But if you really want a thorough deep dive into what pharmacogenomics is, my colleague Aniva, who is an expert in the field, gave a talk yesterday at the Healthcare and Life Sciences Symposium here at KGC. We can all refer to that once the video is going to be available in the KGC portals. In terms of the type of data we need to work with when developing application for pharmacogenomics, there are a number of evidence-based pharmacogenomic guidelines that are available for organizations working to standardize this recommendation. So in the US, most practitioners will be referring to FDA labels for the most common drugs in use there. But there are also other more pharmacogenomic focused organizations and the most prominent one of them is CIPIC, especially in the US. And CIPIC has published numerous guidelines for different drug gene pairs based on existing published research. And a couple of them are shown over here. And outside of the US, other similar organizations, such as the Dutch Pharmacogenomics Working Group, is very active. And there are also other similar organizations in Canada and France that are also shown here. So despite all the rapid advances in genomic sequencing and decreasing costs, translating the pharmacogenomics knowledge in these guidelines into clinical practice has been challenging. As we saw, there are a number of competing standards for pharmacogenomics guidelines out there. This reality, unfortunately, leads to issues like slight variations in interpretation and classification of genetic data across these standards, even for the same patient. What this would practically mean is like one guideline's poor metabolizer might be categorized as an intermediate or some other metabolizer in the different guideline in a different country. And another source of variation in data accuracy can be due to the different capabilities of the labs that perform genetic tests themselves. These are also, or there are also cases where a particular drug is not available in certain countries. So the guidelines will have different drug gene pairs based on that. From our own research and user interviews, we found that clinicians want a tool that understands and incorporates the latest guidelines and standards accurately and automatically. So each time a new guideline is created or an existing one updated, the tool should be capable enough to integrate those without the clinician themselves having to do that. They also want to ask further follow-up clarifying questions about the recommendation that is provided to them by the application. And this can be queries to get, for example, in-depth details on the implication of these recommendations that are shown to them. or they can also simply be a list of the most relevant publication for the particular recommendation for them to verify. So to fully realize the potential of pharmacogenomics-based precision medicine, we need more effective ways to represent and reason over the complex web of relationships between the genes, drugs, diseases, and other patient factors. And that's what we've been building here at Datum. So the Datum platform is an end-to-end biomedical platform to build and deploy secure biomedical applications using both private and public data. And its foundations are built on data-centric architecture powered by Semantic Graph Technologies. So the diagram here shows an overall high-level view of the architecture that we are using. At the center is a pharmacogenomics knowledge graph that is built using publicly available knowledge bases. The data pipeline here is built to ingest structured and unstructured data from this knowledge bases, which are then mapped into semantic concepts into RDF triple stores using our in-house tool. So the choice for the database as a RDF triple store was because RDF triples were the right abstraction that felt our particular use case requirements. And I'll talk a bit more about this choice later on in an upcoming slide. Another important component here is a no-code tool that we built for clinical expert, which is used to encode the decision-making process of pharmacogenomic experts in the form of flows or state machines. They are also internally represented as RDF data and is able to integrate with the knowledge in the pharmacogenomics knowledge graph as well. The flow graph also powers downstream applications that the platform generates, which I'll be showing a little bit later. Another way of looking at our approach for building the Datum platform is through what we call publish, a create, publish, and evaluate model. And this model helps us streamline our thinking when building what is inherently a complex system in a more principled way. So every component of the platform is built to enable one of these three functions. So let's look at the create function that encodes the biomedical knowledge from different public sources into the knowledge graph. As I said earlier, we chose RDF as our knowledge graph representation, as it fits our needs perfectly. We wanted a standardized way of representing the biomedical knowledge in our system, and RDF, which is part of the semantic web standards, fits that requirement perfectly. And another advantage of using RDF we found is the flexibility it gives us in modeling a complex domain like pharmacogenomics, as it does not enforce any schema on the data model upfront. And the ability to define like common set of identifiers as URLs or IRIs for the same concepts across the different data sources also greatly simplified data integration for us without the need for a lot of data transformation in the earlier data pipeline itself. The screenshot on the right shows a glimpse of the knowledge graph we built for a particular pharmacogenomic guideline. So this guideline is for the HLA-B gene and the abacavir drug, which then links to different recommendation and genes across different data sources. And we are also heavily leaning on UMLS, which is widely used in the healthcare domain as the underlying vocabulary that all other data sources can link to. And another component of the create function is the flow builder or the no-code tool, which we built to encode expert knowledge and the decision-making process as flows or state. The interface itself allows linking related pharmacogenomic concepts from our UMLS-based knowledge graph. So the flow is already enriched with knowledge of the pharmacogenomic concepts, which can be really useful for search and AI application downstream, which I'm going to talk about a little bit later. One thing additionally to note here is that the evaluate function is also part of the platform itself. So our tools allow like multiple users to create and evaluate these flows collaboratively. So it ensures the flows are thoroughly vetted through what we call expert in the loop feedback mechanism. We also allow the users to preview the flows in the flow builder itself to get a better sense of how it will work after being published. As this also acts as a great evaluation tool while the flow is still in the process of being drafted. And finally, these expert vetted flows can be published by authorized clinicians, and they are made available to the end users as app in their own private what we call app hubs. So an example of an app that has been published from the flows into the app hub can be seen here. So this application is a very simple source-based application that allows users to find and execute those published flows. And here a clinician can search for a particular drug that are looking to prescribe to a patient. And the flow for the chosen drug is then made available in the app view, in this case as a short questionnaire. You can then quickly input the patient genetic info in the questionnaire to get a recommendation and a nicely summarized report. I'd just like to add that this is only a sample application, and the same application can also easily integrate with an EHR system or a genetic lab testing reporting system to input this data directly. Additionally, we've also enabled the exact same flow to power a question answering LLM interface that we are all familiar with these days. So this AI agent is also able to answer further clarifying questions about the recommendation because the underlying flows are already enriched with the pharmacogenomics data from the knowledge graph. And to get to the right recommendation from free text-based input that the users enter into the chat-based interface directly. We handle the problem of hallucination in the LLM to show the correct recommendation to the users by grounding the LLM output with the facts we pull from our PGx Knowledge Graph in a deterministic way while the flow is being parsed itself. So this is an area we're actively exploring, and we have plans to create even more powerful AI agents for pharmacogenomic use case that can be deployed in real world scenarios. And finally, we also provide an API on top of the knowledge graph for developers if they want to. For example, like we can see an example of an API that allows developers to work with the underlying UMLS knowledge graph itself. And this also fits nicely with the FAIR principles of developing systems that make data findable, accessible, interoperable, and reusable. And so to finally, to conclude the talk, I'll briefly go through the upcoming enhancements for the platform that we've envisioned next. First, we'll continue to refine our use case and are looking to partner with more healthcare systems, labs, and specialty pharmacies who are interested in pharmacogenomics. And we're also looking for other similar use cases in healthcare to develop similar applications. The evaluation function is also a priority for us to be able to deploy fully compliant applications that the clinician can fully trust to be part of their daily workflow. The knowledge graph itself will continue to grow and evolve as we integrate more and more data sources and increase the usability to other use cases as well. And we've also already started fine-tuning certain LLM models to automate the flow creation component itself that is part of the create function as an AI co-pilot, along with other LLM-based work that we're doing with the question answering agent. So this will allow us to scale the knowledge creation process even faster in the future. And, this brings me to the end of my talk today. Thank you very much. And again, I really appreciate the Knowledge Graph conference for giving me this opportunity to present our work here. And yeah, that's it for my talk here.
Thank you so much. Questions from the room? You mentioned that you are working with maybe multiple drug interactions. Should I go there?
Yeah. Sorry. Can you?
Can you hear me now?
Yeah.
Okay.
I can hear you.
You mentioned that you guys are working with maybe multiple drug interactions. Is this upcoming, or did you start working on that in multiple drugs?
So we've already started working on that, but that's still under active development. So just a note, like so for interaction like that. Well, we've been playing around with our whole flow representation to allow different linking between different flows. So that's a more complex use case that we found. So that's under active development, but that's something our partners have been asking for. So we plan to do that really soon.
Okay, sounds good. Thank you so much. Any other questions from the room? All right, I think we're time. Thank you so much for joining us, Saurav.
All right, yeah.